Am I audible?
Yes, sir.
Okay.
হ্যাঁ স্যার শোনা যাচ্ছে।
শোনা যাচ্ছে তো? ঠিক আছে। So in today's class, I'll revise about what are the things which I had taught yesterday. I'll revise it a bit and then go to the new one. So one thing which we have learned is that in all cells of the body have the same genomic complement. So whether it be cells of the kidney, whether it be cells of the liver, whether it be cells of the lungs, whether it be cells of the eye, cells of the skin, all somatic cells. It's better to write the word somatic, not your germ cells. All somatic cells, they have got the same genetic complement. That is, they have the same set of genes. But then the question, and this is known as the concept of genomic equivalence. And that is because it all starts from one fertilized cell called zygote, and that divides and then differentiates to form many cells. So they all have the same set of genes. But then, why is a kidney cell different from that of liver or that of eye? Why are the cells different? The answer to that lies in the concept of differential gene expression. So what is unique over here is that kidney cells, they have one set of genes specific for kidney. The liver cells also have got one set of genes specific for liver. So the set of genes which are for kidney, they may not be the same. Obviously, many of them are not the same as that of liver. So some set of liver specific genes, they are inactive in kidney, and kidney specific genes are inactive in liver. So this is differential gene expression. Ultimate outcome of gene expression is the expression of the protein. Kidneys have got a different set of proteins being expressed. Liver has got a different set of proteins being expressed. Nerve has got a different set of proteins being expressed. So this is actually the differential gene expression. Now, there are some postulates of differential gene expression which we will understand. Is that every somatic cell of an organism contains the complete genome as established in the fertilized egg. We have seen that. Then you have the unused cell in a differentiated cell are neither destroyed nor mutated. They retain the potential for being expressed. That is, the liver specific cells, liver specific cells in the kidney are not destroyed. Even though they're inactive, they're not destroyed. And if you use some chemicals or process and you can activate them, they will definitely become active then. Then the next point which we have studied is that only a small percentage of the genome is expressed in each cell. So out of 100% of the cells, you can only find maybe 1% of the cells are expressed only. Say, for example, 1% of cells are expressed in particular cell. So 100% of the genome is inactive. So these are the three postulates. Now, where is this differential? How is this how is gene expression regulated? Now we have to state how gene expression is regulated. The answer is that is we are talking about. Now let us add one more term. How is gene expression differentially regulated? So essentially the regulation is at 4 levels. First step of regulation is at the level of transcription. Differential gene expression at the level of transcription. In Drosophila, when you study Drosophila sex determination, you will find that there is a process known as alternate splicing. I will now explain briefly what alternate splicing is. I will just open the whiteboard. Okay. So let us think about a gene. This is exon 1. This is intron 1. This is exon 2. This is intron 2. This is exon 3. So this is briefly a six. So when RNA processing takes place, normally we know that this processing can be like, you can have exon 1, exon 2, exon 3, and this gets translated to a particular, translation takes place to a particular protein. The introns are spliced out. You can say that the introns are spliced out. That is this intron 1 specifically, intron 1 and intron 2 are spliced out. Result is you have exon 1, exon 2, exon 3, one after the other. This produces a particular type of protein. Now it can very well be that this part can get spliced out. Intron 1, exon 2 and X. In that case, when it is processed, you have exon 1 and exon 3. Exon 2 and intron 1, exon 2 and intron 2, they have been spliced out away. This could be a such scenario. The other scenario can be that Intron 2 is not spliced. Another scenario can be intron 2 is not spliced. So only this intron is spliced. So result you can have exon 1 exon 2 intron 2 and exon 3. This is another form. So this will form another protein. This will form a larger protein, a different type of protein. So all of these A, B, and C. So A, B, C are alternate transcripts of each other. Am I clear? বুঝতে পেরেছো?
হ্যাঁ স্যার।
কি না
হ্যাঁ 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 বোঝা যাচ্ছে।
বোঝা গেছে, very good. So at the level of transcription, this can be different. Next is another thing which I will sort of another thing which I will be drawing, that is at the level of transcription. There are certain transcription factors. And these different and these transcription factors are needed for the transcription of genes. Then different set of transcription factors in liver, another different set of transcription factor in kidney. So different set of transcription factors are present. You know, one thing you may have studied, that transcription factors bind to promoters of genes on some DNA sequences known as cis-regulatory elements. And what they do, they help to recruit RNA polymerase 2 and transcription takes place. So, if in a liver cell, one set you won't find the kidney set of transcription factors, and so they won't bind to the promoters which contain cis-regulatory elements, because there is no transcription factor, and therefore there will be no transcription of kidney specific genes in liver. Since there is no kidney specific transcription factors in liver, So genes whose promoter contains cis regulatory elements for kidney specific transcription factors CFs they will not be expressed. Clear, Patta? This one?
Yes, sir.
Okay.
Has sir.
Okay. Now we will go to back to the main place where we were studying, the main PDF. So we see that differential gene transcription, however, we have seen two mechanisms of gene expression at the level of transcription. The next is selective nuclear processing regulates which of the transcribed RNA or which parts of are able to enter the cytoplasm and become messenger RNA. We had seen that the RNA is processing. So there is another level of transcription, which is at the level of the RNA processing. At the level of RNA processing. I have to try to delete this part. So the next level is at the level of RNA processing. What are the RNA processing? First thing is splicing. Here also you can see an alternative splicing can take place. A different type of transcript can be produced, right? You can have, you can think, imagine certain transcripts, different type of transcript is present. Sometimes you find an intron coming in and a stop codon comes in. So at the level of splicing, you can have one RNA processing problem. Poly A tail is not very small, so the mRNA, when it enters into the cytoplasm, they get attacked by exonucleases, and then they sort of the transcript gets destroyed before it gets translated. One thing can happen. And the next level is the selective messenger RNA translation. There are sequences known as Shine-Galvano sequences. In prokaryotes and Kozak sequences in eukaryotes. They help in strong ribosome binding. stronger the ribosome binding, stronger ribosome binding to mRNA, this results in better translation and more products are formed. This is one level. The next you can find is differential protein modification. So proteins get modified. There are different types of modifications like acetylation of protein, methylation, phosphorylation, sumolylation, ubiquitinylation. So there are different types of ways by which proteins are modified, and this affects the activity of these transcripts. And these, in fact, eventually result in these modifications of these proteins. They result in differential gene expression. So these are the
Sir,
levels.
my
Yes.
board is not visible, sir.
Huh?
Sir, the board isn't visible.
It's not visible.
Yes, sir. It isn't.
Okay. Okay. Is it visible now?
Yes, sir.
Yes, sir.
Yes,
It's
it is
visible.
visible.
Okay. So I'll repeat this once more. There are sequences. The third point. There are sequences known as Shine-Dalgarno sequence or Kozak sequence. They are present in the messenger RNA of Shine-Dalgarno sequence are present in prokaryotes. Kozak is present in eukaryotes. Is it visible now?
Yes, sir.
Yes, sir.
Okay. Shine-Dalgarno sequence is present, and you have Kozak sequences present. And these are present in the messenger RNA. Shine-Dalgarno is present in prokaryotes, and Kozak is present in eukaryotes. Now, what are the function of this? Allows stronger binding of mRNA to ribosomes. This allows for better translation. This allows for better translation. The next modification level is protein modifications. So in different cells, the proteins which are expressed, they can be modified in different manners. Important modifications are acetylation, methylation, ubiquitinylation, phosphorylation. There are many modifications which happen. Especially important are the ubiquitinylation. They tag the protein for destruction. Acetylation, sometimes we will study right now very soon about acetylation. What can acetylation do? The next thing which you study is methylation. It's important for condensing chromosomes. Phosphorylation of proteins can make the protein active, and dephosphorylation can make it inactive, for example. And so this particular phenomenon, because of a different set of genes and enzymes present in different cells, you can have different genes being expressed. This is another way by which you can see that the genes are differentially expressed. So that kidney cells have got different set of genes and liver cells have got different set of genes. These are many of the reasons by which such a phenomenon takes place. It's actually even not a phenomenon. It's a universal truth that all cells have got the same DNA, but all of them are not expressed. Different sets of genes are expressed in different cells. There are overlapping genes also, but you have unique set of genes being expressed in kidney, unique set of genes expressed in liver. They have also got overlapping same set of genes too. And because of this differential gene expression, and you have a different type of cells and their properties, they are different. Now let us get back to, and now one thing I would like to mention that this differential gene expression and genomic equivalence it was first observed in polytene chromosomes. They were first observed in polytene chromosomes. Now, what are polytene chromosomes? In Drosophila, in salivary glands, there are particular type of chromosomes. They look something like this. So what happens is that when a chromosome replicates, the DNA doubles and they separate away. But in polytene chromosomes, they double, but they don't separate. So 2 becomes 4, 4 becomes 8. And like this, they become thick and they are easily observed under microscope. Now, there are different staining techniques in polytene chromosomes, which you can see. And by these staining techniques, you can see there are certain bands are present. And in certain regions, you can find some puff-like structures present. Puff-like structures present. So let us have a look at these puff-like structures. So this is a polytene chromosome. This, what you see over here, puff, and these regions, this is a polytene chromosomes. You can see certain bands over here and little puff-like regions, these puff-like regions. These are regions of active gene transcription. Active gene transcription. Now, what are these? Hello? Hello?
Sir, audible, sir, but the whiteboard is not visible.
What is the screen is not visible, right?
Yes,
Yes, sir,
screen
it
and
is
also
visible.
you.
My voice is breaking.
The voice is clear,
Yes,
sir.
Screen is not visible, right?
Yes, screen and also your profile is not also visible.
My video, I've closed my video because I'm anticipating a network problem. Therefore, I've shut down my video.
Oh,
So there is a network problem going on. And once of all, I see that Zoom is not very reliable. I've purchased a subscription, but it's not good. So I would rather from next class, we would go on Google Meet. Okay. Anyway, so let's share the screen once more. Let's see what happens. Again, there is a network problem going on. You people stay in the meeting. I will leave the meeting and I will join again, right?
okay, sir.
okay, sir.
Just hold on for a moment. Okay, am I audible?
Yes, sir.
Yes,
Yes,
Okay.
sir.
So these are regions of active gene transcriptions. Uh, just hold on. I will.
Sir, I think you are not audible.
একটা network problem হচ্ছে. I will stop this class right now. Okay. I will again take it properly next day. I will start from this point.
Okay, sir.
কি হচ্ছে?